Hi, hello everyone, welcome back to my YouTube channel, and this is the 10th video on cell signaling. And in the previous video we were discussing about the signaling molecules, and mainly there are 5 types of signaling molecules as you can see here. So the classification is like this, that is lipophilic signaling molecules with intracellular receptors. Second one, lipophilic signaling molecule with plasma membrane receptors. The third one, water soluble signaling molecule, gaseous signaling molecule and other signaling molecules. And in the previous video we discussed about lipophilic signaling molecule with intracellular receptors. Now in this video we will discuss lipophilic signaling molecule with plasma membrane receptors. So we're talking about the second type of signaling molecules, okay, that is lipophilic signaling molecules with plasma membrane receptors. In the next video we will discuss about other things. Okay, now we will focus on this. And if you didn't watch about this lipophilic signaling molecules with intracellular receptors, then better go and watch that video, so that will be helpful to understand the content, the other concepts as well, if you are interested. Now let's begin with the second type, that is lipophilic signaling molecules with plasma membrane receptors. Here these signaling molecules possess plasma membrane receptors. Means here the signaling molecule will be located on the plasma membrane itself. So think this is the plasma membrane, or think this is the animal cell. Okay, I'm hoping you are able to see it. Okay, so the think this is the animal cell. So animal cells have the plasma membrane, right? So think this is the plasma membrane. Okay, this is plasma membrane, and this plasma membrane you can see the receptors. Okay, so these okay, you can see the receptors over here. So these signaling molecules possess plasma membrane receptor. The plasma membrane will be having the receptors. I mean to say, the receptors will be located on the plasma membrane for these signaling molecules. Okay, and here the presence of these plasma membrane receptors confirms the following advantage. It has the more advantage when compared to the other type of signaling molecules which possess the intracellular receptors. Okay, the other rest of the things. How? So what are those advantages? Here there are two advantages mainly. The first one is diversity of end targets is greater. Okay, in case of lipophilic signaling molecule with intracellular receptors, their diversity is less. Okay, here diversity of end target will becomes the greater. So we already know this. What are the different types of end targets? There are mainly 4 types of end targets, that is transfer protein, cytoskeleton, enzymes and genes, right? So in this case, in lipophilic signaling molecules with the plasma membrane receptors, in this case all the 4 types of end targets can be regulated. So thus these signaling molecules shows both genomic and non-genomic effects. Okay, so what is here genomic and non-genomic effect? Here the genomic effect means, here for example I am displaying a picture here, you can see here. So you will get the difference between genomic and non-genomic effect here. Okay, so here what happens in case of non-genomics, so receptors will be there. The signaling molecules send the information to the receptors, then receptors accepts it, and then it will transfer it to the signaling mediators, and then a signal transduction is going to take place and reaches the end targets like effectors. But in case of genomics, what happens? It should go through the translocation and transactivation. It should reaches to the nucleus, and then the process is going to start, that is signal transduction, and then end targets, and then effectors, signaling mediators, and then end target. So it's a long process, so it takes the more hours and days, but non-genomics it takes less time. Okay. These signaling molecule shows both the genomic and non-genomic effect. So the effect will be more here because it's expressing both the ways, that is genomic and non-genomic. And in comparison, when we compare with the other signaling molecules, like signaling molecules with intracellular receptors, that shows only genomic effect. Okay, so that's why diversity of the end targets will increases, so that the effect is also going to increase. That is effect, so that effect is going to increase, so the diversity of end target is greater. The second one, the very important one, that is signal amplification. What is signal amplification? What is, first of all you tell me what is amplification? Amplification means multiplication, right? There is an increase, there is ample opportunities, means there are many opportunities that we can say, right? So in this case, in case of lipophilic signaling molecules with plasma membrane receptors, in this case of plasma membrane receptors, signaling mediators, okay, so signaling mediators facilitate transfer of information from receptor to end targets, and this allows signal amplification. Here what happens, the plasma membrane receptors, here this plasma membrane receptor, okay, this plasma membrane receptors, okay, these are the plasma membrane receptors, right? This plasma membrane receptor facilitate the transfer of information from receptors to end target. Think here is the end target, okay, and here the signaling mediators, we can get the signaling mediators here, right? So here the signaling mediators, so ಇಲ್ಲಿ signaling mediators facilitate transfer of information from receptor to end target. So this allows the signal amplification. What is the meaning of amplification? Amplification means you will get many ways to reach the goals. Okay, you have to reach this end targets. So you can go by this way, you can come by this way, you can come by this way, come by this way, like this. So receptors are here and end target is here. While reaching up to here, you can get many ways, right, like that. So in this case, but if I have to make you understand very easily, compare with the intracellular receptor, signaling molecule with intracellular receptors. So in that case, intracellular receptor signaling mediators such as secondary messengers are not involved. Okay, there are no secondary messengers are involved. Okay, hence signaling molecules with intracellular receptors do not show much signal amplification. But here you can get many signaling mediators like secondary messengers, etc., like that. Okay, but in intracellular receptors you are not going to get that. Okay, so this is the important one. And coming to the examples, so the signaling molecules of this class includes exonoids. Exonoids are of different types, as I mentioned here. Have a look here. The first one, prostaglandins, represented in short form PG, prostacyclins PC, thromboxanes TX, leukotrienes LT, hepatoxins HX, lipoxins LX. Okay, so these are the exonoids. And here, and then this is the signaling molecule here. So where does this exonoid come from? So the precursor, okay, precursor of exonoids is a 20 carbon hypothetical polyunsaturated fatty acid that is called as prostanic acid. Prostanic acid is the precursor of exonoids. Okay, in case of other animals. In case of humans, okay, in case of humans, the precursor is 20 carbon polyunsaturated fatty acid called as arachidonic acid. This is the important acid, very important acid, that is arachidonic acid. Here this arachidonic acid is found in the membrane lipids. Okay, it is going to present in the membrane lipids, attached to the second carbon of the glycerol, which is going to attach to the this is the glycerol. It is attached to the second carbon of the glycerol. It is attached to the second carbon of the glycerol. And moving on to the next point, the enzyme is called as phospholipase A2. This phospholipase A2 cleaves the arachidonic acid. Here, if you take the structure of the glycerol here, the glycerol have two chains here, fatty acid chain. Okay, the second carbon, for the second carbon the arachidonic acid is going to attach, and that second carbon is cleaved by an enzyme called as, very important enzyme, that is PLA2. Okay, it is going to cleaved by PLA2. Okay, that is phospholipase A2 cleaves the arachidonic acid. Moving on to the next point here, that free arachidonic acid then drives the synthesis of ecosanoids by two pathways. Which are those two pathways? There are two pathways, that is one cyclic, or it is also called as cyclooxygenase pathway. Second one linear or lipoxygenase pathway. Okay, so by these pathways the ecosanoids are going to produce. Which are those ecosanoids? Ecosanoids are of different types are here. Those are prostaglandins, prostacyclins, thromboxanes, leukotrienes, epoxytins and lipoxin. So these are the ecosanoids. Okay, these are derived from the from two pathways. Okay, that is cyclooxygenase pathway and lipoxygenase pathway. And how these pathways are going to form? These are going to form from the arachidonic acid arachidonous cleavage, which is cleaved by the phospholipase A2, and this arachidonic acid is present in the plasma membrane. Okay, plasma membrane. That is membrane lipids. So for make you to understand, I have drawn the chart here. So I am hoping this one will help you to understand very easily. So the MM represents membrane lipids. Okay, membrane lipids. I think this is the membrane lipid. Here this membrane lipid. In this membrane lipid, the arachidonic acid is present at the second carbon of the glycerol. So if think this is structure of the glycerol, it is present at the second carbon of the glycerol. Okay, and there the phospholipase A2, the enzyme is going to cleave this arachidonic acid. Okay, then this chain is going to get free. So then that will releases the arachidonic acid, means that the free arachidonic acid is going to form, and this free the released arachidonic acid then drives the synthesis of ecosanoids. Okay, these are the ecosanoids. Okay, in two pathways, that is cyclic cyclooxygenase pathway and linear lipoxygenase pathway. In cyclic cyclooxygenase pathway, the three ecosanoids are going to form. That is first one PG, that is prostaglandins, second one prostacyclins, third one thromboxanes. In linear or lipoxygenase pathway, the another three ecosanoids are going to form. Those are leukotrienes, hepatoxins and lipoxins. Okay, another three ecosanoids are going to form. So this is about the lipophilic signaling molecule with plasma membrane receptors. In the next video, we will discuss about the other signaling molecules, that is water soluble signaling molecule, gaseous signaling molecule, and other signaling molecules. Okay, I'm hoping this video will be helpful. If you learn something new from this video, please do share and subscribe. Thank you everyone.